Antibodies deal with extracellular forms of pathogens and their toxic products. Antibodies, which were the first specific product of the adaptive immune response to be identified, are found in the fluid component of blood or plasma and in extracellular fluids. Because body fluids were once known as humors, immunity mediated by antibodies is known as humoral immunity. Antibodies are Y-shaped molecules whose arms form two identical antigen-binding sites. These are highly variable from one molecule to another, providing the diversity required for specific antigen recognition. The stem of the Y, which defines the class of the antibody and determines its functional properties, takes one of only 5 major forms or isotypes. Each of the 5 antibody classes engages a distinct set of effector mechanisms for disposing of antigen once it is recognized. The simplest and most direct way in which antibodies can protect from pathogens or their toxic products is by binding to them and thereby blocking their access to cells that they might infect or destroy. This is known as neutralization and is important for protection against bacterial toxins and against pathogens such as viruses, which can thus be prevented from entering cells and replicating. Binding by antibodies, however, is not sufficient on its own to arrest replication of bacteria that multiply outside cells. In this case, one role of antibody is to enable a phagocytic cell to ingest and destroy the bacterium. This is important for the many bacteria that may are resistant to direct recognition by phagocytes. Instead, the phagocytes recognize the constant region of the antibodies coating the bacterium. The coating of pathogen and foreign particles in this way is known as opsonization. The third function of antibodies is to activate a system of plasma proteins known as complement. The complement system can also be activated without the help of antibodies on many microbial surfaces and therefore contributes to innate as well as adaptive immunity. The pores formed by activated complement components directly destroy bacteria, and this is important in a few bacterial infections. However, the main function of complement, like that of the antibodies themselves, is to coat the surface of pathogens and enable phagocytes to engulf and destroy bacteria that they would otherwise not recognize. Complement also enhances the bactericidal actions of phagocytes. Indeed, it is so called because it complements the activities of antibodies. And that's antibodies deal with extracellular forms of pathogens and their toxic products. Have a great day.